Well, thank you for inviting me. As you can see, well, I know that forensic entomology is a really hot topic in this department, and as you can see from the title of my talk that my lab has its own version of forensic entomology. And despite what this slide shows, we're not really investigating this sinister guy carrying the sickle and chopping down the wheat. We're investigating a much more stealthy character and its evil activities. We are, let's see, gotta have this right way. We are investigating this guy right here, ugly face, has many aliases known as dead red, flaxseed, the Hessian fly, or a gall midge. It's a juvenile delinquent. It does its damage mostly as a first instar larva, and it's very easy to spot, except that it's really little. It's only a third of a millimeter long, but they're red, and so they're very unusual. And here's the crime scene. I mean, it's total devastation. You can see complete murder, injury to the victim. It's a terrible thing. The innocent victims are quite often susceptible, sweet little wheat seedlings that lack Hessian fly resistance genes. And the evidence that you see at the crime scene are stunting, plant death, which causes thin stands in the field, low yield, and poor seed quality that is reduced by at least 2 US market grades. The modus operandi is that these evil creatures lurk at the base of wheat seedlings. They're in unruly gangs, usually of relatives, and they relentlessly attack at the crown of the seedling. And you can see here some of these guys caught in the act. The murder weapons are kind of unusual. Unlike most caterpillars, these guys don't chew their victims to death. Instead, they use these tiny little mandibles to stab the epidermal cells of the victim. And then they salivate in those cells with poisonous saliva containing effectors that end up manipulating the victim to produce gall-like nutritive tissue, which I'll talk about a bit later. And all of this results in enslaving the victim to deliver nutrients directly to the larva until the victim dies of exhaustion. But fortunately, we have a brave team of breeders, geneticists, entomologists, and molecular biologists who are working to uncover the mysteries of this interaction in our crime lab. So today I'm going to talk about resisting attack by the Hessian fly. And first I'll talk about protecting the victim, which translates as breeding for resistance. Then I'll talk about some strategies that survivors have used, which is really resistance mechanisms. And then we'll talk about disarming the attacker, which is really the effect of resistance on the Hessian fly, and then future directions. So first I'm going to talk about breeding for resistance. So why do we need to breed for resistance? There are over 33 Hessian fly resistance genes known, but unfortunately the Hessian fly is very clever at being able to defeat these genes once they're deployed in wheat cultivars. So currently there are only about 6 genes that are effective in the southeastern United States. But this new gene that we've discovered, which we very cleverly named something really catchy, H33, resides on chromosome 3A on the short arm of wheat, and it confers resistance to all of the Hessian fly populations from the southeastern United States that it was tested against. And so it's a great new gene, and we're hoping that it will be deployed in a smart way, and so that means that we want to tailor the resistance and the release of cultivars specifically to regions of the United States where those genes will be the most effective, and also pyramid these genes with other resistance genes to increase durability or possibly to combine them with transgenic resistance. So now let's talk about these resistance mechanisms. There are, our lab has studied both resistance and susceptibility, but we find that studying susceptibility helps us to understand resistance. And so there are two broad processes that the Hessian fly larvae must alter in the plant in order for it to survive. It has to induce the plant to provide some kind of protection while the larvae develop, and it has to establish a nutrient sink so that larvae can acquire nutrients. So now let's talk about that kind of physical protection that the larvae induce the plant to produce. And the Hessian fly is a gall midge, and so here is a typical insect gall. It's caused by a galling wasp. And so the outside layer of a gall has defense compounds in it. The next layer, the cortex, is antimicrobial. This sort of rubbery area on the inside, this is the sclerotic protective layer. And then the inside surface of that is nutritive tissue, which is specialized cells whose function is to deliver sugars and nitrogen and all the nutrients that the Hessian fly, that the insect larva could possibly want, right to the insect. But the Hessian fly is kind of a minimalist when it comes to being a gall midge. It has found that it doesn't have to work so hard to make a whole gall. All it does is cause the plant to stunt, and that's sufficient to protect the larvae from weather and predators as it develops. And so if you look at this wheat plant, it's infested with Hessian fly larvae down here, and you don't see anything that looks like a gall there. If you peel off the leaf, you see these happy little larvae and no gall there. And even once the larvae get older and are in the second instar, there's really nothing that looks much like a gall. So in essence, the whole plant becomes a gall, and the nutritive tissue develops just on the leaves that are down there in the bottom of the plant, and that's all there is to the Hessian fly gall, and it just develops among those leaves like that. Did I go the wrong way? No, I didn't. So now let's talk about establishing the nutrient sink, so I'm going to talk about inducing permeability in the plant. So in this experiment, we stained seedlings with neutral red, which is a small molecule that can pass inside cells if they have little holes in them caused by permeability. And down here in the control, you can see that plants that are not infested with Hessian fly are not permeable, so they don't take up any of the stain. But these susceptible plants, from 1 day after hatch through 8 days after hatch, they start out with permeability and ability to translocate the stain around the vascular system, and then as time goes on, the entire base of the plant becomes involved, and so everything essentially becomes permeable. And if you look at the plant in cross-section, 2 days after egg hatch, the leaf on which the larvae reside takes up stain and is permeable in the epidermal cells and in the vascular bundles. And then a day later, this permeability has spread to little developing leaves inside the whorl of the plant that don't even host Hessian fly larvae. So the permeability spreads in 3 dimensions in the plant. If you take a close-up look at permeability and nutritive tissue formation, this comparison shows that a normal epidermal cell of the wheat leaf has a nice big healthy nucleus, and the cells form a flat surface on the leaf. But once the cells become converted to nutritive cells, the nucleus starts to degrade and sort of shrivels up in the corner here, and the cytoplasm acquires a lot of organelles, including Golgi and mitochondria, and these things pump out nutrients and energy and all the stuff that Hessian fly larvae need to develop, and they fill the plant so full of stuff that it bulges out and is really turgid, and eventually it just pops and the stuff in here leaks out and the Hessian fly larvae consume it. So now let's talk about resisting permeability. I showed you the progression of permeability in susceptible plants, but resistant plants also become permeable. This shows one day after hatch that there's a little bit of permeability in this resistant plant, which is right where the larvae are, and as time progresses it becomes a little more permeable, but then things seem to go backwards and the holes get plugged up and the plants are no longer permeable. So this suggests that there's something different going on in resistant plants from susceptible plants. This slide shows the structure of the epidermis of a wheat leaf, and these are epidermal cells here which are fortified with cellulose and lignin and lignans that make the cell walls strong. But then on top of that is secreted a layer called the cuticle, which contains waxes and other kinds of lipids, including cutin, and that's a protective layer that helps conserve water inside the plant, and also some of the waxes repel insects. But there are many genes that are known to be involved in the synthesis and deposition of these compounds. So first I'm going to talk about cellulose. And so cellulose is in the cell walls of the plant cells. And when we did our experiment to measure the amount of cellulose, we found that in susceptible or resistant plants, which are always going to be blue in this talk, the amount of cellulose is about the same as in the control plants, but in susceptible plants, the cellulose degrades, and so 4 days after egg hatch, you can see that there's less cellulose than in the resistant or the control plants. And the decrease in cellulose may contribute to the thinning of cell walls in susceptible plants and the eventual lysis of those cells. We also looked at the expression of dirigent genes, and these are genes that mediate the formation of lignins and lignans, which are some other compounds that fortify the cell wall. And we found that this gene, the messenger RNA abundance, increases very rapidly 1 day after egg hatch in resistant plants, and then decreases slowly over time. And so this message is much more abundant in resistant than in susceptible plants. We also looked at the expression of lipid transfer protein genes, and lipid transfer proteins help to maintain the cell integrity by moving the cutins and the waxes and all the lipids to the surface of the epidermal cells. And when we looked at the expression of one of these genes over time, we found that the resistant plants and the control plants had about the same amount of lipid transfer protein messenger RNA as time progressed, but susceptible plants had less and less of this message, and it's as if the gene is being silenced by virulent larvae infesting the susceptible plants. So then we also looked at a lot of, well, 10 different genes that are listed here that are involved in the synthesis and the deposition of those lipids, the cutins and the waxes, to the surface of the leaf. And in the resistant plants, all of these genes either had the messenger RNA level either stayed about the same, or sometimes the message became more abundant in resistant plants. But in susceptible plants, all 10 of these genes looked as if they were becoming silenced over time. So the insect is turning off mechanisms that maintain the integrity of the leaf surface. So then we did a gas chromatography experiment, and the data here's shown 4 days after egg hatch, and certain waxes, so these are all wax monomers down here, and in the resistant plants, certain wax monomers became more abundant in the resistant plants, but not in the susceptible plants. And this is important because it's a protective layer, and sometimes waxes can repel certain kinds of insects. And then we looked at cutin, which is another lipid, this is 1 day after hatch and 4 days after hatch, and these are all Cutin monomers. And several of these Cutin monomers were degraded in susceptible plants, but not in resistant or control plants. And so there are mutants of Arabidopsis where these Cutin-associated genes have been silenced or turned off, mutated. And in those cases, the plants become more, the epidermis of those plants become more permeable. And so that's makes us think that these may be involved in permeability in wheat also. So I already showed you this panel with the progression of permeability in susceptible and resistant plants. And we started looking at when defense molecules are being made in the plant, and found that they're expressed at about the same time that the plant is the most permeable. And here we are showing, these are Western blots, so this is protein that we're looking at that's made by 2 different lectin genes, and it's made by resistant plants but not susceptible or uninfested plants. And it's made right at the time when the plant is the most permeable. Also, the plant makes, reactive oxygen species. So these are things like superoxide and hydrogen peroxide that are, and peroxidase is involved in generating these compounds. And so these become more abundant at about the same time the resistant plants become more permeable. And these can function to signal to the plant that they can be signaling molecules to help induce other resistance mechanisms, or they can also be ingested by insects and work as feeding deterrents. Then we looked at some genes that well, actually, we looked at the levels of polyamines in the wheat plant and in the Hessian fly. And polyamines are stress signaling regulators, or they can also function to scavenge reactive oxygen species like peroxidase, or hydrogen peroxide and superoxide, and which would then make the plant, the susceptible plant, more hospitable for the larvae. And so the concentration of these polyamines increases in susceptible plants but not in resistant plants. And at the same time, the larvae that were living on these susceptible plants also have more polyamines in them. And so it's unclear to us as to whether both the plant and the insect are ramping up their expression of polyamines to deal with stress, or if possibly the larvae are obtaining some of their polyamines from their plant host. So susceptible plants deliver nutrients to the virulent larvae, and we decided to look at the production of free amino acids, since those are nutrients that the larvae need. We used an amino acid analyzer experiment and found that these amino acids listed here became more abundant in susceptible plants. And these 2 amino acids are particularly interesting because phenylalanine is an essential amino acid that these insects must obtain from their host, and proline is an amino acid that is stored as an energy source for insects. So these are the most highly abundant amino acids in susceptible plants. But resistant plants are able to turn down the production of these amino acids, especially these ones that are needed the most by the insect. And so the resistant plants are able to deprive the larvae of some of the nutrients that they need. So now let's talk about global characterization of changes that lead to plant resistance and susceptibility. And so these are lots of people who are in my lab or have been in my lab who have worked on these kind of projects, and this is one of our collaborators. We did a microarray experiment where we dissected out about the bottom centimeter of the little developing leaf that the larvae were living on, and we did a time course of 8 days and looked at resistant and susceptible plants compared to uninfested controls, and to see what genes were responding to Hessian fly in the plant. And so we noticed two main things. That there's a timing difference between resistant and susceptible plants. So in resistant plants, the biggest response to the Hessian fly is the first day after egg hatch, and then things sort of calm down after that point. Whereas in susceptible plants, genes get either upregulated or downregulated right away, but as time goes on, more and more genes become involved in converting that plant to just the perfect environment for the Hessian fly to develop. So and also the number of genes involved was quite different between resistant and susceptible plants. You can see here that there are many fewer genes that we were able to detect in resistant plants than in susceptible plants. And so this indicates that susceptible plants go under a massive reprogramming of their plant developmental processes so that they can really cater to the needs of the Hessian fly, whereas resistant plants are very efficient in depriving the larvae of what it needs and getting the job done really fast. And so it's a very energy efficient kind of resistance. We also did a laser capture microdissection experiment, and in that one we sectioned tissue and looked for places where we could find larvae sitting on top of the leaf, and then used a laser to cut out a few cells that were right underneath the larvae, and we made our tiny little RNA preps out of these little pieces of tissue, and then did a microarray experiment and compared the laser capture microarray experiment to the whole tissue microarray experiment that I talked about in the previous slide. And we found that although in the whole tissue microarray there were a lot more genes detectable in susceptible plants, when you look right at the feeding site, we found more genes detectable and responding to Hessian fly in the resistant plants than in the susceptible plants. And although the whole tissue and the laser capture experiments had some genes in common, there were really a lot of genes that we could identify by laser capture that we just missed with the whole tissue microarray. So that indicates to us that the broad area where the larvae are residing has a resistance response, but there are things that happen right where the larvae are that are important and that you can't detect. So that indicates that at least some aspects of resistance are very localized. The laser capture microarray allowed us to identify genes that we couldn't find in the previous microarray experiment, and so here are a few of those genes that were interesting to us. In the susceptible plants, we identified genes that in other plants have been linked with gall formation, and we couldn't find those at all in the whole tissue microarray. And then we also, in susceptible plants, found certain genes involved in glycolysis and TCA that were active early in the laser capture experiment, but couldn't be detected until much later in the whole tissue experiment. And then in resistant plants, there were several disease resistance-like genes that we found that we couldn't detect in the previous experiment. And one of them is a gene that's like a leaf rust resistance gene. So it's one of these receptor kinds of genes. And this one is also a receptor kind of resistance gene, both of which were quite high upregulated in the laser capture material right at the feeding site in resistant plants. So now let's talk about consequences of resistance to the Hessian fly. You gonna change? Change. How do I change it? Okay, there we go. The larval midgut is often a target for resistance because when the larvae ingests plant liquids, it ends up in the midgut, and so if the plant can target the midgut, that's an easy way to mess up the insect. And so one set of molecules that does this are lectins, which I talked about in a few slides previously. And what these do is they bind to the inside surface of the lumen of the larval midgut. And they bind to glycoproteins that are found on the surface. And by doing that, they can disrupt the microstructure of the midgut. And what they tend to do is affect the microvilli, which are these little projections that greatly increase the surface area of the midgut so that the larvae can absorb food. And so this experiment shows cross-sections of larval midguts 3 hours after egg hatch. And in the first square is a susceptible midgut and a larva on a susceptible plant. So it's been feeding there for 3 hours, and the microvilli are intact, and you can see that there's contents in the lumen. Then the green rectangle shows the cross-section of a larva that never fed on a plant. It has been put on moist filter paper for 3 hours, and you can see that starving on filter paper doesn't degrade the microvilli. But larvae that had been on resistant plants for 3 hours, their microvilli were greatly affected. So the microvilli start to become misshapen or swollen or completely destroyed, as you can see here. And so this is only 3 hours after egg hatch. So resistance gets induced quite rapidly and has a very fast effect on the larvae. So if you look at larvae 24 hours after they've been on a resistant plant, the microvilli are completely gone and the lumen contents has been evacuated. So this really looks like quite a stomach ache to me. And if this video would work, but it doesn't work unfortunately, you would see this poor larva sort of writhing around. And this is a larva that has been on a resistant plant for 24 hours, and it doesn't act at all like a larva on a susceptible plant, which by this point has settled into a permanent feeding site. So how do these larvae die? Well, in order to survive, the larvae have to be able to induce nutritive tissue formation by continuously feeding on a susceptible plant and applying saliva for 4 days. And if that doesn't happen, the plant doesn't stay permanently stunted and doesn't become a really resistant plant. But Hessian fly, avirulent Hessian fly larvae don't feed for very long. They stop feeding by about 24 hours after egg hatch. So resistance may be a combination of cell fortification, the delivery of toxins, which would then disrupt the larval digestion and deter it from feeding, and also deter it from continuing to interact in the same way with the plant. So the larvae may just die a slow death of starvation over 3 to 5 days because the plant doesn't do anything that just kills them off right away. They hang around for quite a while. So this is really a low-cost kind of defense by the plant. It doesn't have to invest a lot of energy, and plant pathology books are always talking about how high-cost resistance is for the plant, but it's not in this case. In fact, a resistant plant that has been infested with avirulent larvae has no yield penalty, so it makes the same number of seeds and the same quality of seeds as a plant that has never been infested by Hessian fly. But there's always a conspiracy theory. And so in this case, the virulent larvae conspire to rescue their avirulent siblings. And this happens when avirulent and virulent larvae co-infest the same plant. So here's the virulent and there's avirulent larvae. And the way that we think this happens is that the avirulent, even if the avirulent larvae get there first and they induce resistance, when the virulent larvae arrive, they induce susceptibility, which results in this spreading permeability, and eventually permeability spreads under where the avirulent larvae are, and so then they have access to the nutrients. And although they would have a damaged midgut, they must be able to regenerate that because in co-infestation experiments, avirulent larvae can survive if virulent larvae are present. So even if resistance is induced first, systemic induced susceptibility can override localized resistance. So let's talk about some future strategies for new ways to resist attack by this serial killer. One thing that we need to do is really identify genes that are essential for resistance and susceptibility. And so one way we're doing that is by doing viral induced gene silencing experiments, which turn off genes, and so then you can see whether the plant is more or less resistant with a gene turned off. So the first gene we've tried is SGT1, which is required in order for many of these receptor-like resistance gene cascades to be effective in plants against other pathogens. And so we had 7 plants that were silenced to various degrees, and so if the level of expression of this gene is 100% in a non-silenced plant, these silenced plants had various degrees of silencing, varying from about 50% silencing down to about 2% of the level that the control would have of this. And so then we looked at the size of the larvae on these same 7 plants. And this is the unsilenced control. And you can see that on all of the silenced plants, the larvae are bigger than on the control plant, which indicates that the silenced plants were a little bit more susceptible. Yes, a little bit more susceptible than the non-silenced plants. And although these differences aren't big, they were highly significant. And in this experiment, we didn't manage to get the plants silenced before the larvae hatched because the timing is very difficult on these experiments. So we're going to repeat it a few more times and see if we get the plants silenced earlier, if we can see even a bigger difference. Another thing we're doing is we're cloning promoters that could be used to drive transgenes in new resistance strategies. And we're also trying to figure out different parts of those promoters that may be specific for making the gene respond to Hessian fly attack. So far we have cloned 5 promoters. We've gone 2 kb upstream of the open reading frame, and all 5 of these genes share 14 promoter motifs. And so some of these are kind of interesting. One of them is a binding site for a worky transcription factor, and this particular type is involved in upregulating defense genes in rice that are induced by elicitors from pathogens. So that's an interesting promoter motif that we'll be pursuing. And 3 other promoter motifs are involved in dehydration response and are found in genes, other kinds of genes that respond when a plant becomes dehydrated. And we found in our microarrays that several dehydration genes responded to Hessian fly. So those are also motifs that would be interesting to follow up on. And then there was a binding site for a transcription factor that influences the level of salicylic acid inducible response in defense genes. So that's something else that we want to follow up on. So these promoters that we're cloning, we have sent to our collaborator, Roger Tilmany, who has transformed plants for us. But we were very specific about the kind of promoters that we wanted. We wanted things that were highly upregulated by Hessian fly larvae and that responded very quickly within the first 24 hours of the attack, because if they're driving an insecticidal transgene, we want it to affect the larvae before susceptibility has been permanently induced. And then also these promoters had to turn on their genes at the larval feeding sites, but definitely not in the seed, because if we're attaching toxins to the promoter, we don't want it in the part of the plant that is going to be consumed by people. So far Roger has used 3 of the promoters and has them driving Gus, which is a reporter gene, in wheat and brachypodium, and we have those seeds back from him and are about to start looking at experiments that will help us see exactly where the genes driven by those promoters get expressed in wheat. And we're using our new Illumina RNA-seq experiments to identify additional promoters that we would like to use in transgenics, and also to help us identify other promoter motifs that might be important. So if we're going to have promoters, we have to have something to hook up to promoters. And Shuba has developed an assay. It's called a HIT assay, and right now I can't remember exactly what that stands for, but I might think of it in a minute. But in any case, the point of this assay is you put the protein that you want the larvae to ingest into the liquid at the bottom of a test tube, and put the plant in there, and it translocates the protein up to where the larvae are feeding so that they can ingest it. And we have to do this because the larvae are obligate parasites and can't survive on artificial diet like Drosophila can. And so we tested several lectins and a proteinase inhibitor and a Bt toxin. And one of those lectins, Galanthus nivalis agglutinin, was the most effective against Hessian fly, but also the cryotoxin was pretty good. And so then we looked at GNA-treated larval midguts and found that the microvilli were becoming destroyed just as they were when the larvae feed on a resistant plant. So these plants are susceptible, except that they now have the protein that we have translocated to the larvae. So this lectin acts by destroying the larval midgut microvilli, and also the larvae 7 days after egg hatch are about the size of newly hatched larvae, whereas their cousins that lived on untreated plants have gotten to be much larger. So in summary, I have talked about how the Hessian fly rapidly defeats resistance genes that are deployed in the field, and that we identified a new Hessian fly resistance gene that is effective in the southeastern United States, and that it's one of only 6 resistance genes that are currently useful in that region. And we want to be able to protect these resistance genes so that they will last longer. And so we're looking at new strategies of resistance to combine with native resistance. And in order to do that, we need to understand resistance and susceptibility at the molecular level, because we uncover secrets that we think can be used for genetically engineering new forms of resistance. And so we have characterized the processes of resistance and susceptibility and what things are able to be blocked by resistant plants that happen in susceptible plants. And we're mining several data sets to find promoters that can drive transgenes that can provide new forms of resistance. So I'd like to thank everybody in my lab. This is, Shuba is a staff research scientist in agronomy, and Jill, Andrea, and Chad are or have been technicians in my lab. David, Kurt, and Nagesh have been postdocs. Jason, Marcelo, Stephen, Jacob, Abdulrahman, and Melissa are graduate students, some past and present, and Keely was an undergraduate, and these are some of our collaborators. Thank you.